Okay, hi there. Welcome to another episode of Science and Technology, History of Science and Technology Q&A. This is on a different day than usual. My apologies for switching things around. This is a time of year where we're in the middle of our summer educational programs, and I keep on getting pulled into things related to that. In fact, we'll be live streaming a few of the events probably from our summer school. We have well, 3 or 4, depending on how you count it, different programs all running. Right now, this week, we have a middle school summer camp. Next week, we have week 0 of our summer school. That is the pre-week for the physics track of our summer school. Then we have 3 weeks of our summer school. And in the last 2 weeks of that 3-week period, we also have our high school summer camp. So anyway, lots of educational programs. The middle school one is a little easier for me because we are not assigning individual projects to everybody, and that's part of my responsibility for our summer school and high school summer camp. So that's kind of my moment of extreme professoring in the year when we'll have, I don't know, 100 and something students, and the goal and the thing we always achieve is to come up with unique original projects for each student. And I typically make a list of potential projects, and people pick partly from that list and partly from things they want to do. And then I'm kind of responsible for the final selection there. Anyway, lots of fun for me, but also rather an intense period with rather tight scheduling. So these live streams might bounce around a bit, but there may be some interesting, unique additional live streams, which we'll be doing as part of particularly the summer school. All right, so let's see. There were all kinds of questions left over here. There's one from Mikhail about how was DNA discovered, history behind its discovery. Not sure I can tell you all of the details there. A few of the characters involved I do know somewhat. I mean, Jim Watson is still alive. I've certainly met a number of times. And Francis Crick, I knew somewhat. Those were the sort of 2 most credited people. And I guess Jim Watson wrote this book called The Double Helix, which is kind of a adventure storybook about the discovery of DNA in 1953. I think its first sentence is, "I have never seen Francis Crick in a modest mood," or some such other thing. I'm not sure that that would quite be the way that I would describe at least my interactions with Francis Crick, but of course I knew him much later in his life. There's a little bit of the science of what was going on. I mean, before, people knew that there was DNA in the cell nucleus, and that had been known for a long time. I think people back from the 1920s, 1930s had known that. I don't know how much Thomas Hunt Morgan, who was a big sort of fruit fly genetics person, I don't know exactly how much he knew. I think he knew things about the molecular, what kinds of substances were involved in the cell nucleus, but just how heredity worked wasn't. I mean, it was certain laws of heredity were known. They'd been figured out by Gregor Mendel, and other people had figured out these, heritable traits and these different kinds of dominant recessive traits, all this kind of thing. But the mechanism by which that worked was not clear. And there was this sort of idea of genes and so on, which were all a bit kind of based on the observation of what effects they had rather than the mechanism by which they were made. And so there was DNA, it was in the cell nucleus. It probably had something to do with heredity, but it wasn't really known how that worked. And there had been some vague thinking, I think, that it might be something that was a little bit like kind of storing data or something. But the idea of storing data wasn't yet a big thing, because remember, historically, 1953, which was sort of the big year for the figuring out of how DNA works, that was something that was in the very early days of computers. I mean, today, when you say there's information, there's data, it's stored digitally, everybody would know what that meant, because everybody is used to kind of the world of computers, but that simply didn't exist in those days. The typical, let's say, physicist or something would never have seen a computer. The typical biologist would never have seen a computer. I suppose there was so this idea that there would be this sort of digital information thing wasn't really a thing yet. I'm kind of thinking that, and I may have my history slightly mixed up. There was a physicist named George Gamow who was also much involved in the origination of the Big Bang model of the early universe and so on, who maybe it was after the actual main discovery of DNA, but he was involved in later in figuring out the code by which you go from base pairs to proteins, to amino acids to proteins and so on. But I think that there have been some suggestions that maybe there was some sort of digital information component to DNA. Okay, but then the real question was, there's this molecule. What's the structure of the molecule? So a few other molecules, biological molecules, their structure had been figured out. Normally, how do you figure out the structure of a molecule? Well, the big method that was used was X-ray crystallography. So the thing to understand is, in a crystal, the atoms are arranged in a very regular periodic way. The question is, in what way are they arranged? You have a diamond crystal, you have a salt crystal, the atoms are arranged differently. In a salt crystal, it's a cubic arrangement of atoms. In a diamond crystal, well, it's usually called the diamond arrangement. I guess it's a hexagonal-ish arrangement of atoms. And the question is, what is the arrangement of atoms? And there's this technique called X-ray crystallography, which is a way, given that you have a crystal, it's a way of saying what is the arrangement of atoms in the crystal. And it works by reflecting X-rays from the crystal. And when the X-rays are reflected specifically from the atoms in the crystal, and depending on how the atoms are lined up, you get lines of destructive or not, or constructive interference between the emission of the re-emission of X-rays from the surface. It's actually very much like the way that a phased array radio works. When in 5G, for example, one has this whole idea of, one will emit radio from this sort of distributed antenna, and it will be all directed in a certain direction because the phases of the different things coming from those different parts of the antenna will be arranged so that the radio energy goes in a particular direction. It's sort of the same kind of idea with X-ray crystallography, except that there's sort of a natural array that is made up by the atoms in the crystal lattice. And so that's kind of the way that one tries to figure out how a molecule is arranged, how the atoms in a molecule are arranged is, well, if you can make a crystal of that molecule, with that molecule, then everything is lined up, many copies of it are lined up, and then you can use X-ray crystallography to try and find the arrangement of atoms. Now, one of the big challenges there is how to actually do the crystallization, how to take this material and have it form into a big crystal. And that's a very tricky thing, and the sort of theory of crystallization is still quite incomplete, and it's still kind of often something of a mysterious art to make a big, perfect crystal. It's something very important in the semiconductor manufacturing, making these big crystals, big as perfect as possible crystals of silicon. But back in the day, people were making crystals of some of these biological molecules. I think hemoglobin was an early one. I think myoglobin was another one. I seem to remember the chap who was, I think it was a person called John Kendrew, who was the person who decoded, I think, myoglobin by crystallizing it and then using X-ray crystallography. Strangely enough, in the weird connections of this world, he was an alumnus of the elementary school that I went to in Oxford, England. And I remember there was a sort of a grand sort of science exhibition opening thing when I was like 8 years old, that this then very ancient seeming person showed up to, and I at least said hello, so to speak. I don't know. That was the that shows the difference in timing. So that was that will have been 1968 or so. The person who had sort of decoded myoglobin was by that point an older person. In any case, the big challenge in all these things was how do you crystallize these complicated molecules? Because if it's a complicated molecule, it's flapping around all over the place. It's not easy to make sure that it can sort of line itself up perfectly. But that was kind of a big challenge. I don't know the full history of this. I think Rosalind Franklin in London was the person who was most deeply involved in kind of the crystallization and X-ray crystallography of DNA. But the question was, when you do X-ray crystallography, what you get is a bunch of bright spots from what were the angles at which the X-rays were kind of emitted from the crystal. And then the question is, that gives you essentially a Fourier transform of the actual distribution of atoms. In other words, what it's telling you is, if you look at if there was a periodic array of atoms with a definite period, that would be like a wave of a definite frequency, and that would be like a single spot. But what you're seeing is what arrangement of different periodicities of atoms are there going in different directions, and that's the arrangement of spots that you get. And so there's then a big decoding problem. If you've got this arrangement of spots, now there's sort of the puzzle of, well, what's the actual arrangement of atoms that gives that particular arrangement of spots? And in general, it's not unique. In general, for any arrangement of spots, and technically it's because you only have the intensity, you don't have the phase information of the X-rays, you only have the intensity of the spot in a certain angle. But the question is, given the intensity of spots, given this distribution of spots, what is the actual structure of the molecule that produces that? And as I say, it's not unique. So there could be many different shapes of molecules that could produce the same distribution of spots. Of course, many of those shapes of molecules would not be plausibly constructible out of atoms. And so then there's this kind of almost mathematical problem of, can you kind of reverse engineer the thing to figure out what structure would lead to this pattern of X-ray crystallography output? And I think the dynamics of what happened is, I think Rosalind Franklin was the person who got the X-ray crystallography done. And I think then Jim Watson and Francis Crick were the people, they were working in Cambridge, England. They were the people who figured out kind of what would be the distribution of atoms that would lead to that X-ray crystallography pattern. And they famously had this thing made out of Meccano that was kind of this helical structure that was the thing that they figured out would lead to that pattern of spots. And so that was sort of the big thing, was figuring out that structure. And then the question was, well, so how does that structure actually work? What does it do? And that took a number more years. And I think in the original paper that they wrote in Nature magazine in 1953, they had sort of a couple of sentences at the end saying, this might suggest that perhaps there's a whole sort of digital information story associated with DNA, which I think kind of implies that they weren't the ones who originated that digital information story, but nobody could kind of see how to make it work before it was known what the structure of the DNA molecule was. Now, I think it's sort of interesting to see the kind of career tracks. I think Francis Crick had been doing physics before kind of going into this sort of biology direction. I mean, in his later life, he kind of changed direction and worked on things about the brain and neuroscience and consciousness and so on. I met him when he was in that sort of phase of life. Jim Watson continued very much in the molecular biology, was a co-author of a sort of standard textbook, was very much involved with Cold Spring Harbor, which is kind of it's a place with a very unfortunate history that goes back to the world of eugenics in the early 1900s in the US, but is a distinguished place where a lot of important research in molecular biology has been done. He was also involved, Jim Watson was also involved in kind of the pushing for a government-sponsored human genome project and so on. He's also a person who I would say believes that perhaps the things that he can deduce about the world from science are definitively the way the world has to be, and sometimes I think perhaps downgrades sort of other views about the world and comes to all kinds of conclusions that he says, look, it's scientifically provable that this is true, but actually he's sort of missing a big piece of the picture. But that's kind of his subsequent life. I mean, the thing for which he is best known is the 1953 discovery of the structure of DNA. So that's kind of the story there. And of course, it took a number of years. It wasn't, well, I see the history from far after it happened, but by the 1960s, things were really hopping in terms of the development of molecular biology. As a field, molecular biology kind of started popping up as departments in universities only considerably later, 70s, 80s, and so on. But as something where people were sort of figuring out the key ideas, figuring out messenger RNA, transfer RNA, how that whole sort of mechanism worked. That was really a story of the 1960s, centered around places like Cambridge and Cold Spring Harbor and places like that. And I guess Jim Watson was at Harvard for a while, so it was kind of the two Cambridges and other places. I think one person who was a